Yesterday we were discussing B cell activation, and we have already discussed one of the process in which the B cell is activated, that is TH dependent. So can you guys just tell me what happens in TH dependent B cell activation? First of all, the pathogen is going to come then.
Sorry, doctor, I can't see your screen.
Hello, can you guys see my screen?
Yes.
No.
Yes, we can.
I can
Okay.
see.
Okay. Hello, can you guys hear me? Hello.
Yes, we can hear
Yes.
you.
Okay.
Yes, we
So
can
I
hear
was
you.
asking that we yesterday we discussed B cell activation, that is TH dependent, right? So can you guys just repeat the phenomena? Like first of all, which steps are going to be involved in activation of B cell, that is TH dependent? First of all, pathogen is going to come. Then what happens?
Peptide antigen.
Okay, perfect. It would be peptide. Okay, then your B cells is going to engulf it, right?
Yeah.
It is going to present it on which receptor?
MHC class 2.
MSC2. Then what happens?
T cell receptor
Okay,
CD4 plus.
helper or killer?
Helper.
Helper.
Okay, CD4 positive. You also have a co-stimulatory receptor as well. What's that?
B7 CD28.
Yes, CD28. Okay. Okay. So now let's discuss the concept of affinity maturation. So what is affinity maturation? So as the name indicates that it's like related to some kind of affinity, right? Okay. So whenever the B cells are activated, can you just tell me the place inside the cortex where the activated B cells are present? Okay, perfect. There are germinal centers. So it means activated cells are going to be present in germinal centers. What do we mean by activated B cells? It means they would be actively producing antibodies, right? Plasma cells. And they would be producing antibodies. And these antibodies are going to react with their respective antigen, right?
Yes.
Like for example, if I have an antigen who has like their code of 231, okay, another I have an antigen who have like an 123 of code, let's suppose. Okay, I'm just giving a random example. Then I have another antigen that is 321. So your B cell is going to produce antibodies, millions and billions of antibodies. Once they produce an appropriate pattern of antibodies, for example, it would be producing millions, like okay, it would be combining their like genetic material in order to have millions and billions of variant of antibodies, right? Once it produce specific 3 to 1, so now it's going to bind it well with this, right?
Right.
Okay, so this would survive. So the concept of affinity maturation is that your antibodies are changing these variable structures pattern in order to have best affinity towards its respective pathogen, right? Is the concept okay? For example, this 3 to 1 is going to survive because they would be having best affinity. Okay, they would love each other. Okay, otherwise like it would be producing millions and billions of different patterns, right? And once the pattern matches and you have highest affinity, those antibodies are going to survive. Those who do not have good affinity, okay, or they are random, they would die, they would apoptose simply, right?
Right.
Okay, then we already discussed the concept of isotype switching. What is isotype switching? You guys know? Just tell me. B cells, which receptor? MHC2, right? It's going to combine with CD4, right? Can you just tell me the co-receptors, co-stimulatory receptor?
CD40 and
CD40,
CD40 ligand.
40 ligand. Then you are going to see class switching, right? Okay, can you just tell me the first antibody that is being released by your B cells with the
IgM.
IgM? Perfect.
Yeah.
So this was the concept of B cells activation. Now let's discuss antibodies and its structure. Guys, like for example, this is your B cell and you have B cell receptors, which we have discussed. It might be CD19, CD20, the upper limit numbers, right? So basically, this individual receptor is same as that of antibody. Yesterday we have discussed both. Simply, it's Y structure, right? Okay, so you have two regions of antibodies. You guys may know that is your FC portion and FAB portion. So this would be your FAB portion, and this would be your FC portion, right? So This FC portion, okay, you have a terminus here which is known as C terminus, okay? Okay, so this is in opsonization. What's opsonization, guys?
Coating the pathogen with antibodies so that it can be easy for phagocyte.
Yes, like
Phagocytes.
okay, yes, like simply it's tagging the antigen in order for the immune cells to come here and eat it up, right? It's known as opsonization. It's attracting your simply your immune cells, right? So this FC portion is like responsible for the opsonization, right? Okay. Similarly, the IgG and IgM can also bind to this FC portion. Okay, like in complement system, we will be discussing the how there are certain pathways for activation of your complement system. One of which is by activation of IgG and IgM, right? So then you have Fab portion. You know the Fab portion here you have variable region, right?
Yes.
And it's highly specific for an antigen. As we have already discussed, you have several patterns. For example, 1, 2, 3. Okay, let me call it VDJ genes, right? Like VDJ genes, you can just relate it with the 1, 2, 3 concept which we have discussed here. They like keeps on like changing their patterns in order to have like many of the patterns of your immune like antibodies, right?
Right.
Okay, so isotype switching. Okay, now let's discuss IgM. You know IgM is the biggest of all, right? It's basically a pentamer. Okay, pentamer it means you are going to have five antibodies attached to one another. Okay, via a chain. What's that chain called?
J. J.
Yes, it's J. Okay, so basically, okay, do you guys know the concept of affinity and avidity? You have an MCQs in your world. You guys might have practiced that MCQs as well. Hello, hello. Can you guys hear me?
Yeah.
Okay, so basically, what is affinity? Affinity is like the love for this variable site to bind to a pathogen, right? So basically, the affinity of IgM towards pathogen is less, but its avidity is more. What do we mean by avidity? The total size for attachment of this pathogen is more.
More.
You guys can see, like there are 10 sites simply. So its affinity towards pathogen is less, but its
Avidity is more.
avidity is more, right? Okay, so basically, whenever we have like a parasitic infection, so IgM is going to release as a result of that. Then it's the first uh, antibody which is being released by your B cells, right?
Right.
Okay, then you have IgG. So you guys just tell me, what do you know about IgG? It's the most abundant immunoglobulin in your body, right?
Yes.
Most abundant, and it's just a monomer, single Y, right? Hello.
It's a dimer, I guess.
Yes.
Okay. Sorry, it's a dimer. Sorry. So it's a dimer basically. Okay.
Is
So
it?
IgA is dimer.
IgG is monomer.
IgE is
dimer.
monomer,
Sorry,
guys.
sorry.
IgE, don't confuse me, please. I believe you guys. It does not mean that you make me wrong.
Sorry, Dr. Remy.
Okay. So it's a monomer. Okay. Like now I have just gotten old, so I believe you young guys. So I thought that you were correct and I was wrong. Okay. So it's basically monomer, right? It's the abundant immunoglobulin inside your blood. And for example, the initial immunity to child is provided by IgG. Why? Because it's a monomer anchor cross placenta, right?
Right.
Yes.
So one of your high yield point, you guys are going to see an MCQs related to it. The only immunoglobulin which can cross your placenta,
IgG.
IgG. So it means for the first 6 months of immunity of your baby, it's going to be provided by your IgG, right? Okay.
Yes.
IgG is also a good option in, right? Here we have discussed IgG and IgM is going to activate your complement system. It's going to act as an option in, right? Okay. Then we have IgA. We have discussed IgA is going to provide immunity to which structures?
Mucosal.
Mucosa.
Mucosa.
Okay, mucosal. Perfect. It's going to be present in malt. What is malt?
Mucosa
Mucosal
associated
associated lymphoid tissue.
lymphoid tissue.
The Pire patches, which we have discussed. It's simply a fancy name for the Pire patches. Okay. In breast milk, it's also present in breast milk, right? And it's dimer.
Yes.
Yeah, it's a diamond.
Okay. So, and it's attached via J chain, right? It's your J chain. Okay.
Okay.
Okay. Now let's discuss IgE. You guys know IgE related to mast cells. So you guys are going to tell me, we have already discussed the details of mast cells while we were doing asthma, right?
Yeah.
First of all, what happens? Can you just tell me asthma is type TH1 response or TH2 response?
H2.
H2.
Okay, if
Yes,
it's
true.
TH2 response,
TH2.
it means your B cells are going to be activated first. They are going to go, they are going to engulf this like allergen. They are going to present it into your T cells, CD40 ligand. You are going to say isotype switching, IgE is going to be formed, right? IgE is going to come, they are going to attach itself on the mast cells, right?
Right.
Then if you have re-exposure, you are going to see aggregation of these IgE or cross-linking of this IgE, and ultimately you are going to see histamine being released, right? So it's a Th2 response, like it play important role in your type 1 hypersensitivity IgE, right?
Yes.
Okay, so then you have IgD. IgD is also a monomer, okay, and it isn't quite significant. So now let's discuss immune complement system. So you guys are going to tell me what's complement system. Okay, what do you guys know about complement system?
Innate immune response.
Okay, it's an innate immune response. Okay. Okay, it has three types. Can you just name those?
Classical.
Classical, alternate, and lactic.
Alternate, classic, and lactin.
Active.
Okay, so you have certain components of the complement system that is from C1 to C9, right?
Yeah.
Yes.
Okay, but they aren't activated in this manner. That's first of all C1, C2. Like they are just, there are some like random activations, and ultimately you are going to see a complex from C5 to C9, which is known as membrane attack complex, right?
Attack
And
complex.
membrane attack complex is going to go, it's going to destroy your pathogen simply.
Yes.
Okay, so first of all, let's discuss the types of complement system, and then we are going to have discussion on. And I will be just writing down some key points. You guys have only just, you guys are only going to remember those. Those are your high yield points. First of all, let's discuss classical pathway. So what happens in classical pathway? Can you guys tell me?
Antigen antibody
Antibody
reactions with
binding.
IgM and IgG.
Okay. Okay. So the
C1
main
binding to
concept of, yes, the main concept of all of these pathways in order to produce C3 convertase, right?
Hmm.
And convert. Okay. So these components are basically released from your liver in an inactivated state. Once they are activated, they are going to convert into CA plus B, right?
yes.
Okay. This is general for all of the components, right? So the main goal of all of these three pathways is in order to activate your C3 via an enzyme that is C3 convertase, right?
Hmm.
C3 when converted is going to be converted into C3A plus
A and B.
C3B,
Hmm.
right? So now let's discuss classical pathway. First of all, for example, a pathogen enter your body. First of all, what happens? Your IgG and IgM are going to tag them, right?
Yes.
Okay, then what happens? Your complement system, like it can, you can just imagine of an opsonization. Your complement system is going to come here.
Yes.
Okay. First of all, your C1 is going to be activated. C1 complex, right?
Yes.
Yes.
Okay. C1 complex is going to activate your C2 and C4,
C4.
right?
Hmm.
Once C2 is activated, you know, whenever they are activated, they are going to convert into A and B,
A
right?
and B.
So it would be converted, C2 is going to be converted into C2A plus C2B, right?
Yes.
Right.
C4 is going to be converted into, okay, sorry.
4A, 4B.
Okay, it's going to be converted into C4A plus C4B, right? So
Yeah.
your C2B and C4B is simply your C3 convertase, right?
Okay.
Right.
So what's
Yeah.
the role of C3 convertase? It's going to activate your C3, right?
C3.
C3 is going to be converted into C3A
A and
plus C3B. Okay, ultimately C, C2B, C4B and C3B, all of them collectively will form C5
5
convertase.
convertase.
You guys can just draw this picture if it's difficult for you guys to remember, okay? C5 convertase is simply going to activate your C5, right?
Hmm.
Into C5A and C5B.
Hmm.
C5B is going to give you simply the membrane attack complex, right?
Yes.
Yes.
Okay, so doctoral futures, can you guys give us an overview how do the different component of your complement systems are activated? Okay, 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 perfect. Okay, C3 convertase, yes, yes, yes. Yes, it can simply just activate your membrane attack complex. So this was basically your classical pathway, right?
Yeah.
Yes.
Okay. What's your lectin pathway? Only the components which activates your complement systems are changed. Like in this case, your IgG and IgM was activating your complement system, right? But in
Yes.
lectin pathway, what happens? You have
Carbohydrate.
MBL. What's MBL? Mannose binding lectin. It's one of the component of your innate immune system, right? It's going to bind to mannose or the carbohydrate parts of a pathogen, simply mannose.
Yeah.
And ultimately, it's going to activate your C1, simply. Then all of the reaction is common.
Yes.
Okay. So can you, Dr. Renu, can you just tell me what's the difference between lectin and IgM and IgG pathway or classical pathway and lectin pathway?
Yeah, so in the classical pathway, the antibodies like IgG and IgM are binding to the pathogen that is
Okay.
activating the C1 complex. But in
Okay.
the lectin pathway, we have this MBL, that is mannose binding lectin, and it
Yes.
binds to the carbohydrate
Portion that
proteins
is
of the
mannose sugar.
mannose sugar from the antigen.
Yes.
Yeah.
Okay.
So that
So now let's
activates the
And
just
C1 complex. Yeah.
here
yes.
C1 complex is not required. It can directly stimulate C2 C4.
Okay. It can you mean to say that it can directly activate your C2 and C4.
Yes. Yes.
Okay, yes, like it formed like like this complex is basically similar to that of C1 complex. It does not activate your C1 complex. Like it's similar to C1 complex, right?
Right.
Yes. Yes.
It can just drive this pathway simply. Okay. So so now let's discuss alternate pathway. What happens in alternate pathway, Dr. Ramesh?
Uh, there's a auto activation of C3 to.
Perfect. So the C3 is basically auto activated. Some of the C3 is auto activated. So you are going to see C3A plus C3B, right?
C3.
Okay, then this C3B is
But
going to join with a factor, bind with a factor that is also one of the component of your innate immune system. That is your factor B,
B.
right?
Yeah.
B and A.
Factor B. So once the C3B and this factor B binds, it's simply it forms C3 convertase.
Okay.
Okay. Yes. C3 in this case, C3B binds with factor B.
B. Yeah.
It's going to form, this is your factor B, this is your C3B. Some auto
Yes.
activation of your C3. It's going to form C3 convertase
Yeah.
simply,
Yes.
and ultimately you are going to see formation of your activation of your complement system, right? And membranotic complex, simply.
Yes.
Okay. So inside our cells, in order to, like, for example, this is my cell. Let's suppose my normal cells. So in order to, like, survive against a complement system, all of our cells have some components, some inhibitory components. Do you guys know what's that?
DAF.
Okay, that's DAF. DAF. It's present on all nucleated cells. So in order to, like, save these cells from your complement activation, your normal cells, it's like, you can say it's the immunity of your normal cells against your complement system, right? Okay, why is it significant? For example, whenever we are sleeping, so what happens? Basically, our respiratory rate is decreased, right?
Right.
Yes.
Okay, it means you're going to see CO2 retention, right?
Yes.
Okay, remember, CO2 retention or acidosis triggers your complement pathway. It activates your complement pathway. You guys must remember it, right?
Okay.
Okay. So in order for the normal cells to, like, to escape or to just save themselves from the complement system, they have these immunity, that is like DAF factors present on the surface of their bodies, right? So for example, those individuals who have certain mutation and they do not have DAF, what happens during night time? You are going to see destruction of red blood cells, simply, and that condition is basically your paroxysmal nocturnal hemoglobinuria,
Yeah.
right?
Right.
Okay, we
Yes.
will be discussing the details of this pathology in red blood cells module, right? But here you guys must know the concept. I'll be discussing it there, so you guys should know what happens. Dr. Rino, can you just give us the overview of this pathology? Just a quick overview.
Yeah, so this DAF, it is present on our cell surfaces.
Okay.
So in any acidotic environment, like when we are sleeping, it's actually a preventive mechanism that
Yes,
are
it's a preventive mechanism.
yes. So like in acidotic environment, when CO2 rises, so
Okay.
our complement system will get activated.
Okay.
But yes, so in this there is a deficiency of DAF in PNH.
Yes, there is deficiency, or you guys can say there is some mutation in which
Okay.
you are going to see, like, disorganized or non-functional DF. Simply, we'll be discussing the details, but here you guys must know the concept so that you can relate it there with your with like the complement system concept, right?
Okay.
It's only for relation purposes. Okay, so this was all about the complement system. Now let's discuss the pathology section, right?
I have one question. So, how deficiency of DEF is related to RBC destruction?
Okay, so as I have already discussed, that DAF is like simply a preventive mechanism, as Dr. Renu told us. It's a preventive mechanism. Like, okay, this is going to inhibit your C3 convertase. Normally, your red blood cells, whenever the C3 convertase come in contact with them, they are going to inhibit it. Or they, okay? Are you getting me?
Yes.
Yes.
Okay,
Yes.
so in this way, they would the red blood cells and nucleated cells are going to prevent themselves against your complement system via DAF. Simply, for example, if a person do not have DAF on the surface of their cell, it means your complement system can now attack your cells. So paroxysmal
Okay.
nocturnal hemoglobinuria, especially at night time. Why? Because during night time, respiratory rate is low, you are in acidotic state, and it's very favorable environment for your complement to be activated. So as the condition
Okay, I
name,
got it.
paroxysmal nocturnal during night time hemoglobinuria. When you destroy your red blood cell, you are going to release hemoglobin, right? And it would start appear in your urine. So it's paroxysmal nocturnal hemoglobinuria. Simply. Okay. So now let's discuss the first condition, that is primary immunodeficiency. Okay. Okay. So our first topic of our discussion would be DiGeorge syndrome. What's DiGeorge syndrome, guys? We have discussed it a lot of time, so you guys are going to tell me. And how is it related with this immunology? Hello.
Yes.
Okay, what's Dijor syndrome, guys?
Thymic aplasia.
Okay, third and fourth do not form. It means which structure won't form?
Thymus.
Okay,
Thymus.
if thymus do not form, it means what happens? You won't be seeing maturation of
T
your
cell
T cells, right?
deficiency. T
Right.
cell deficiencies.
Okay, so for example, if a person is having T cell deficiency, so it's very favorable for the virus and fungal infection to happen inside the body, right?
Yes.
Okay,
Right.
so it's simple. So DJOR syndrome is basically the failure of third and fourth. Okay, someone why have like it's very noisy. Kindly you guys can. Okay, thank you. So basically, whenever you have failure of third and fourth pharyngeal cords to form, so what happens? You won't be forming your thymus as well, and as a result of that, the positive and negative selection of T cells cannot happen. So as a result of that, T cells would be deficient, or the like mature T cells would be deficient. So it is very favorable environment for the virus and fungi to invade inside our body and simply attack it. Simply, okay. So other condition which we'll be discussing is severe combined immunodeficiency. Okay, just don't confuse yourself by the name. So basically, what happens in this condition? For example, you know all of your immune cells are dependent on your signaling molecules, right? That is cytokines, right? Right. So, for example, if you do not have cytokines receptor, if a cell is deficit with cytokine receptor, so can you just tell me, would we see proliferation and activation of B cells and T cells?
No.
No. Okay. Secondly, for example, if we have certain deficiencies in MHC class 2, can we see the activations of B cells and T cells?
No.
No.
I do not have class 2. It means my B cell cannot be activated. It won't be interacting with your CD4. I won't be seeing any class switching, right? We already know the basics.
Yes.
Okay. Thirdly, for example, if certain toxic substances in the B cells and T cells build up, so it can also destroy your B and T cells, right?
Yes.
Mm-hmm.
Okay. So what happens normally, like normally whenever the adenosine, what's adenosine?
Purine base in DNA.
Okay, so basically, like simply you can say that ATP is formed of adenosine. Whenever you consume your ATP, you are ultimately going to see adenosine. So adenosine inside your cell love to combine with some of your amines. Simply, I'm just making it very simple, right? So whenever they combine with one another, they are very toxic for your cell. But inside our body, there is an enzyme which do not allow adenosine to bind to amine. That is ADA,
ADA.
adenosine deaminase, right? Okay, if I have deficiency of adenosine deaminase, it means adenosine and amines are going to combine and they are going to destroy your B and T cells, right? So all of these three mechanisms lead to destruction or like non-activation of your T and B cells, B and T cells, right?
Right.
Okay, so this condition is basically known as severe combined immunodeficiency syndrome. If your B cells and T cells aren't functional, okay, if I have certain cytokines defect, I cannot activate my B and T cells. They are of no use. It means I'll be seeing some fungal infection, some viral infection, some bacterial infection, some intracellular organism infection, TB infection, right? And you cannot give a vaccine even to them because vaccine is all related with your T cells, your memory, right? If you give them a live vaccine, it means you are just inserting a bug and causing a disease directly in them, right? Are you guys getting me?
Can you just repeat the last thing? This disease.
Okay, so we were discussing about severe combined immunodeficiency syndrome. So the basic idea is your B cells and T cells aren't functional, right? So what would be the factors which leads to B and T cells malfunctioning? It would be, for example, if this B and T cells do not have cytokines receptor. Secondly, if you do not have any MHC2 complex or MHC2 receptor, right? Thirdly, if certain toxic substances are building within your B and T cells, they would keep on destroying your B and T cells, right? So as a result of that, both of your T and B cells aren't working inside your body. So it's a immunodeficiency syndrome, right? So it's a
Yes.
severe combined. Combined mean both B and T cells are not functioning. Okay? If your B cells and T cells aren't functioning, it means you are going to see bacterial infections, right?
Yes.
Because it cannot form antibodies against them. Simply, right? You cannot, okay, the viral infection cannot be coped out simply because you do not have T cells. The fungal infections. So this individual is prone to like every infection simply. It may be due to bacteria, virus. Okay. So sometimes yesterday we have discussed what's the concept of vaccination. Basically, we give them like their vaccination have certain types. For example, there is a live vaccine in which we just like weaken the organism and insert it inside our body in order to have the T cell memory. If you guys remember, all of the vaccination, the concept is having memory, right? Okay, if they do not have T or B cells and I'm inserting a live organism inside them, so even this weakened organism can form like a severe infection inside these individuals simply. So what's the treatment? Place them in a sterile isolation, also known as bubble baby, or give them stem cell transplant because all of the deficiencies with B and T cells, right?
Yes.
Okay. So the second condition which we will be discussing is X-linked A gamma globulinemia. X-linked A gamma. Like I'm trying to make these pathologies quite simple, so that's why I'm using some like layman terms here.
Doctor, today's class is for how many hours?
Okay, so we are left with 2 to 3 pathologies, so I think it would just take 20 to 30 minutes. Okay.
Okay, thank you.
Thank you.
Okay. So we have X-linked A gamma globulinemia. As the name indicates, you have less gamma globulins, right?
Yes.
What are gamma globulins? Your antibodies, right?
Right.
So it means this pathology is directly dealing with your B cells.
Yes.
You're having some pathology related to B cells, right? Okay. So you know how do the B cells like send their signals? There are certain signaling pathway mechanism. If you guys remember, that is tyrosine kinase. If you guys remember, in case of insulin, right?
Yes.
Okay, like this B cell is going to okay, I'm giving you a simple concept of it. This B cell is going to convert into plasma cell, right? Then it's going to convert into antibodies, right?
Right.
This B cell sends signal to your, like let's suppose nucleus via certain signaling pathway, right? So simply that is tyrosine kinase pathway. Okay, so if I have certain defect in this pathway, it means B cell cannot be converted into plasma cells. It means your antibodies could not be formed well. So you are going to see A gamma globulinemia inside the blood. The gamma globulin would be less, A gamma, or no gamma globulins. Okay, so it's basically an X-linked disorder because simply you can think of the gene for your tyrosine kinase is basically present on your X chromosome. It's also known as Bruton tyrosine kinase. That's X-linked. So for example, if a person is having like deficiency of gamma globulins, it means it's very difficult for this person to fight off bacterial infections, right?
Yes.
But now you guys are going to tell me first 6 months of life, you won't be seeing any problems. Why?
IgG
Because maternal
and
antibodies are there.
Maternal antibodies.
Perfect. IgG, already discussed. Okay, so you are going to see some okay, can you just tell me that this guy would be having some mucosal infection as well? Why? Like upper respiratory tract
IgA.
IgA. If he cannot form plasma
there will
cells,
be
it
no
means he cannot.
isotype switching.
Yes, there won't be any isotype switching. So you are going to see enteroviral infection, bacterial infection, giardia infection. Okay, and the maternal antibodies are going to give them like immunity for the first 6 months. Then they cannot. As the maternal antibody wanes out, then you are going to see the complication, right? So live vaccine can also be avoided in this kind of individual, right? Why? Hello.
It can cause.
Yes,
It
we
can
have
cause severe
already
infection.
discussed. Yes, it can cause infection because the concept of your memory was that B cell activation of TS dependent B cell activation is going to give you memory. right? So you cannot form memory, and you do not have any antibodies to fight off this life organism as well. So now Dr. Al Future is going to give us an overview of both of these diseases, like a quick overview. Just make it simple within 3 to 4 words. They're very easy, I guess. Perfect. No timers. Okay. Perfect. Mainly your viral and fungal infection, right?
Yes.
Okay. 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 Why aren't they not functioning? Okay. Then can we give them live vaccination? Okay. Perfect. What happened? Okay. Good. Okay. What's your X-linked A gamma globulin? Just make it simple. Yeah. Perfect. Okay. So this was all about the pathology. Hope so you guys just understand. Okay. So now let's discuss another condition, that is IgA deficiency. So you guys are going to tell me it's very simple. What happens in IgA deficiency? For example, if a person is deficient with IgA, so what happens? You're going to see mucosal infections, right?
Yes.
Yes, simple. Okay. Yes, it can be definitely caused by X-link. Yes, you can have, but you can also have sometimes isolated IgA deficiency.
Yes.
Okay. Now let's discuss hyper IgM syndrome. You guys know what's hyper IgM syndrome?
Last switching, we only produce IgM.
Okay. So only IgM is produced. What do you think? What would be the basic, like we have already discussed it. What's the basic pathology?
Deficient.
Class switching
Perfect.
defective.
Perfect. If you have, okay, class switching is basically undergone whenever you have CD40 and CD40 ligand come together, right? So if we have some mutated CD40 ligand, it means isotype switching cannot happen simply. So in that case, your B cells is going to dump IgM only. So you are going to see a lot of IgM.
Mm.
Okay. So second signal cannot be delivered. Okay. So you are going to see low IgA. If you are having low IgA, so what happens? Mucosal infections, right? Low IgG. Okay, recurrent infections. Why? Because IgG is an opsonin. You do not have any opsonin, so you can have many bacterial infections, right?
Right.
Okay.
Okay, if you do not have IgE, so we may also see some hypersensitivities. Hypersensitivity cannot take place. Like for example, like Ig is basically produced during parasitic infections or during, okay, so parasites can easily attack and simply, okay. So now let's discuss autoimmune disorders. The first disease which we will be discussing it. Okay, so now first of all, let me give you a brief overview. How do the autoimmunity take place? You guys know the self tolerance. Can you guys tell me what's central tolerance and what's peripheral tolerance? Okay. 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 No, central tolerance was your negative selection. For example, if the negative selection do not take place, what's negative selection? Your CD4 is going to bind with your body antigens.
Yes.
Okay. If you have failure of negative selection, it means some of the T cells are going to escape. They are going to become activated or reactive against your own body pathogen. So it's an autoimmunity. Peripheral tolerance is whenever T cells come with the peripheral body proteins and you have deficient CD28 and B7. Okay, let me repeat it again, guys. Okay, so Dr. L features what happens in negative tolerance. Okay, okay. Let me write it here. So if you have strong connection between your T cells and your own body cells or body proteins, so it means they would die off. Why?
Yeah.
If they survive, it means you are going to say or see autoimmunity, right?
Yes.
Because these cells would be reactive against your own body,
Yes.
right? If due to certain phenomena, due to certain like defects, this negative selection do not take place properly, or these T cells escape this negative selection. So it means now you have T cells that would cross react against your own body proteins simply, right? So it's an autoimmunity. You are activating your own cells against your own body parts simply.
Yes.
So this is your central tolerance. Negative selection is your central tolerance. So it means those cells who escape the central tolerance would have ability of autoimmunity. Or in other words, let me make it simple. If they escape the negative selection, it means they are going to attack your normal body parts. Okay?
Yes.
Did you guys understand?
Yes.
Hello. So why are you guys so low? Just tell me if you didn't understand. I will repeat it again. Hello.
Can you repeat?
Okay, what happens in the negative selection? Okay, in the thymus, first of all, you are going to see positive selection. In positive selection, what happens? First of all, you have a cell that is double positive. What do we mean by double positive? They have CD4 as well as CD8 receptor, right? First of all, in the positive selection, they are going to be exposed towards MHC1 and MHC2. For example, if the CD8 binds well to MHC1, they would survive. If they do not bind well to your MHC1, they would die. Right? If the CD4 bind well to your CD2, they would survive. Oh, sorry, MHC2, they would survive. If they do not bind well to MHC2, they would die.
Yes.
Okay? They will then move towards the second phase. So now here we have those cells who have passed their first exam, right? They have passed their step one. Right? Now they are going to appear in step two, right? So in step two, what happens? You are going to see negative selection. Now these CD4 cells and CD8 cells are exposed towards your own body antigen. Like your antigen presenting cells are going to take different proteins from many parts of your bodies. They are going to express them to your CD4 and CD8. If they bind well, they are going to die because we do not want cells that should cross react to our own body parts or our own body proteins. So they would die. It's negative selection.
Yes.
Okay? If they do not bind well to our own body proteins, they would survive. Right? Then they would come out and simply they are going to be converted into individual CD4 helper and CD8 cytotoxic. Right? So the concept was, for example, if we have certain like defect in expressing these body proteins to these cells, or in other words, for example, if the sum of the CD8 and CD4 cells escape the negative selection, okay,
Yes.
if they come out through this pathway, let's suppose, okay, they did not give step 2 and they directly come here, it means we have certain T cells, CD4 cells and CD8 that would be cross reacting to our own body parts.
Yes.
So what's the fancy name for that? The T cells or the immune cells reacting to our own body parts, it's known as autoimmunity simply. So it means negative selection is a protective way of your body, right, against your or against autoimmunity.
Yes.
So let me give it another name. It's a central tolerance.
Okay.
Okay. So for example, if these cell escape, now they are going to cross react with your own body proteins, right? But the better part is we also have a second mechanism in order to protect ourselves against these escaped T cells. That is peripheral tolerance, right? So we have discussed whenever antigen presenting cells come in contact with CD4 cells, you are going to see MHC2 to CD4, right? And then you have another co-stimulatory receptor that is B7 and CD28, right?
Yes.
It means that these receptors are only present on antigen presenting cells, right?
Yes.
Yes.
The B7, for example, is only present on antigen presenting cells or those cells who have antigen simply,
Yes.
right? But our normal body cells lack these B7, right? Hello?
Yes.
Is that clear? So, for example, these T cells came and they cross react with your body parts. Let's suppose liver. So it means liver do not have B7. Okay, liver do not have B7. So the second mechanism for protection against these autoimmune cell is lack of B7. Though these cells have no B7. So they would tell these T cells that please sleep. Because I have recognized you, you have escaped this process because I do not have B7 and you are supposed to join to those cells who have B7.
Okay.
Okay, so this is your peripheral tolerance. If the did you guys understand?
Yes.
Yes.
Okay, Dr. Al Futures, please come forward. Okay. Yes. So what's central tolerance and peripheral tolerance, Dr. Al Future? Okay. 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 Yes. Okay. Okay. And and it's basically your central tolerance, right? Okay. And B7. Okay. So if we do not on a normal cell we do not have B7, that would just tell those cells that yes, I have recognized you. You have just escaped this process and you are going to sleep now because you just cheated, right?
Yes.
Okay. So now let's discuss systemic lupus erythematosus. A very interesting topic, and I love to teach this topic. So you guys are going to participate as well. Okay. So what's SLE? What do you guys know about SLE? We are going to make it very simple. Okay, we will be using some layman terms. Okay, what happens to my pencil? Just give me a second. Okay. Okay. So now let's discuss SLE. What do you guys think? What is SLE?
It's a systemic autoimmune disease.
Okay, it's autoimmune disease. It means you're going to see certain immune cells cross-reacting with our own body parts, right?
Yes.
Okay. So, what's the idea of SLE? Like, what happens in SLE?
This is an actually early complement deficiency, like C3b.
Okay, so it's an early complement deficiency.
Yes, sir.
Okay, it's an early complement deficiency. Just make it simple. Like for example, if I'm a 10-year-old child, so just tell me what's SAB. Okay, simply it's an autoimmune condition, right? In which what happens, your immune cells are going to cross react with your body cells and it's going to cause destruction, right? And for example, if it's causing destruction in your, like it would cause destruction in your various body parts, and ultimately it would just give you manifestation related to that simply. Okay, so let me give you a, okay, let's start from sunlight. Okay, for example, okay, what happens to my pencil? Sorry, just give me a second. Okay, guys, so my pencil is just ran out of charge. So we are going to continue class just right after 10 to 15 minutes. What do you guys say?
Yeah, that would count as a break. Thank you.
Yes, it would be as
Yes,
a break.
good, sir.
Don't leave the class. And so we're going to
No.
continue.